Hello dear student, I am Rajiv Saxena, Associate Professor, Smriti College of Pharmaceutical Education, Indore. In today's class, we'll discuss about IMViC test. Now, the full form of IMViC test is Indoor Production, Methyl Red, Vogus Proscios and Citrate Utilization Test. This is a very common test which is performed in any microbiological laboratory, especially for the identification of the bacteria that belongs to the category of Enterobacteriaceae. There are two major bacteria that are having the Enterobacteriaceae family, that is E. coli and Enterobacter aerogenes. So if we want to go for the identification, characterization and differentiation of these two categories of the bacteria, then definitely this IMViC test is very essential and important in any microbiological laboratory. So one by one we'll discuss about all these tests and what is exactly the principle of these tests and how this laboratory in a particular laboratory, how these tests are performed. So the first test is a indoor production test. Now, as you can see that in a bacteria, if we are having the nutritional components are available, and as we have also discussed in the segment of various essential nutrients, then we have discussed that in nutrition, first we have to go for the incorporation of various nitrogen sources, like especially your various essential amino acid. So if in a media, if a tryptophan is available as one of the essential amino acid, then the bacteria will have the capability to convert or utilize this tryptophan in the presence of enzyme which is called as tryptophanase in order to produce the indole molecule. This is one of the very basic biochemical reaction that takes place inside the bacteria, that this tryptophan in the presence of enzyme tryptophanase is converted into indole. But this type of arrangement is actually available only with E. coli, but it is not available with Enterobacter aerogenes. So what will happen if in a sample, if whatever the sample which is brought into the laboratory, if only the E. coli is available, and if you are trying to cultivate in a culture media that consists of the tryptophan, then what we can see that only the E. coli will be able to utilize this tryptophan in order to produce the end product which is indole. Whereas in the sample, if Enterobacter aerogenes is available, then definitely this tryptophan cannot be utilized. So the reaction suggests that the tryptophan is converted into indole if we are having as E. coli as our sample. Now, once this indole will be produced, then we need to go for the identification of the indole. If this type of reaction is taking place inside the culture broth, and if we are seeing that the indole is available, indole is produced, then we cannot visualize the indole. But for the visualization, for the identification of the indole, what we have to do, we have to go for the addition of one reagent which is called as a Vogus reagent. This Vogus reagent generally consists of amyl alcohol, hydrochloric acid and para-dimethyl amino benzaldehyde. So once we go for the addition of the Vogus reagent after the cultivation of the bacteria in a broth that consists of the tryptophan, what we will see that the red color ring will be appeared on the top of the culture broth. The formation of the red color ring indicates that in our sample, the bacteria that is available is E. coli and it is not the Enterobacter aerogenes. So this is the first test that is used for the identification and characterization of the bacteria, that is E. coli. So with the help of these tests, we can go for the differentiation of the bacteria, that whether in our sample the E. coli is available or in our sample Enterobacter aerogenes is available. Now, the next test, which is again associated with the identification and characterization of the bacteria of Enterobacteriaceae family, is actually the Methyl Red Test. Now, the Methyl Red Test is basically dependent upon the pattern of utilization of the glucose and the formation of the end products. So, what we can see that in a bacteria, we will go for the addition of the glucose as one of the essential carbon source or the carbohydratic molecule that will help in the growth of the bacteria. But this glucose inside a bacteria will undergoes a metabolic cycle which is called as glycolysis, and the end product of the glycolysis is pyruvate. So this type of reaction will take place in both in case of the E. coli also and in case of Enterobacter erogens also. But what will happen once this pyruvate will be generated in both the categories of the bacteria, but further the pyruvate if we are talking about an E. coli, it will undergoes another pathway which is called as a mixed acid pathway. And because of the mixed acid pathway, the pyruvate is further converted into various end products which are acidic in nature. For example, lactic acid, formic acid, these type of acids are generated because of the mixed acid pathway which is available in case of the E. coli. But if we talk about Enterobacter erogens, then again the glucose will be converted into pyruvate, but further this pyruvate will undergoes another pathway which is called as butylene glycol pathway. But because of this butylene glycol pathway, the end products which are generated are neutral in nature. Whereas if we are talking about the mixed acid pathway, the end products are acidic in nature. Whereas if we talk about the butylene glycol pathway, the end products are neutral in nature. Like for example, 2,3-butybutanediol, acetone, these are the products which are neutral in nature. So these products are generated if the category of the bacteria is Enterobacter erogens. But if the E. coli is available, then definitely the acidic products are generated. So in this situation, if in a culture broth, if we'll go for the addition of one of the indicator which is called as methyl red. And as we know that the methyl red indicator gives a red color in the presence of the acidic environment. But what we can differentiate here, in case of the presence of the E. coli, the acidic end products are generated, and because of the generation of the acidic end products, the pH of the broth or the media will becomes acidic. So when we go for the addition of the methyl red dye, the red color will be formed. Whereas if in our sample, if Enterobacter erogens is available, then what will happen? Because of the formation of the neutral product, the pH will not be shifted towards the alkaline side, and when we go for the addition of this methyl red dye, it will not give the red color. So because of the addition of the methyl red dye, if the red color is observed, then what we can conclude that in our sample E. coli is available and it is not the Enterobacter erogens. So with the help of these two tests, what we are doing? We are trying to go for the identification and differentiation of the bacteria of the family Enterobacter erogens. What we have learned, like if we are having the indole production test, if the indole will be produced, and when we go for the addition of the Vogus reagent, then what will be formed? Then because of this reaction, the red ring will be formed on the top. The presence of the red ring indicates that in our sample E. coli is available. Similarly, if we talk about the methyl red test, because of the addition of this methyl red indicator, if the red color is generated, then again we can conclude that in our sample E. coli is available and it is not the Enterobacter erogens. Now, the third test of the IMViC test is Vogus test. What we can see that it is actually the continuation of this methyl red test. What we have concluded in the last test, because of the formation of the neutral product, the red color is not generated when we go for the addition of the methyl red indicator. But when we see that what are the different neutral products are generated, then we can see that it is 2,3-butanediol, acetone, these kinds of the neutral products are generated. So again, if the glucose is converted into pyruvate and pyruvate is undergoing butylene glycol pathway, if we are talking about Enterobacter erogens, then what will happen when the acidic products, when the neutral products are generated, and in this situation, if we go for the addition of 40% KOH, then what will happen? Especially this acetone will be converted into diacetyl. So what we are concluding here that the Vogus, first thing is Vogus test is a continuation of the methyl red test. In case of the methyl red, we have already differentiated that if the red color is available, then E. coli is there, and if the red color is not generated, then maybe Enterobacter erogens is available. But again, we have to confirm that whether the Enterobacter erogens is available or not, then that can be identified by continuous Vogus test. since the neutral products are generated and acetone is one of the neutral product. So in this situation, if we are not adding methyl red, we are adding 40% of KOH, then this acetone will be converted into diacetyl. Now further, this diacetyl in the presence of the alpha naphthol and guanidine, means we have to go for the preparation of the media that consists of alpha naphthol and guanidine. So because of the presence of this alpha naphthol and guanidine, this diacetyl is converted into some red color end products. So again, if the red color is observed after the addition of 40% KOH and in the presence of alpha naphthol and guanidine, then we can confirm that in our sample Enterobacter aerogenesis is available. Here we are getting the red color ring that confirms the presence of the E. coli. Here we are getting the red color because of the addition of methyl red. Here we are also concluding that in our sample E. coli is available. Okay. But if the red color is not observed, we have to go for the confirmation that whether in our sample Enterobacter aerogenesis is available or not. That can be identified by another test which is called as Vogus test. So what we have to do in this situation, we have to go for the addition of 40% KOH that results in the formation of diacetyl, and further this diacetyl in the presence of alpha naphthol and guanidine, it is giving a red color. So after performing again these two additional steps, if the red color is observed, then we can confirm that in our sample Enterobacter aerogenesis is available. Now the last test of IMViC test is citrate utilization test. Now again you can see that there is one basic biochemical reaction that is taking place, that the citrate in the presence of enzyme citrase, it is converted into oxaloacetate and acetate, and further this oxaloacetate is converted into pyruvate and carbon dioxide. This reaction is somewhat similar what we have learned in the first step, that the tryptophan is converted into indole in the presence of tryptophanase. But again the citrate is also converted into oxaloacetate in the presence of enzyme which is called as citrase. But what is the observation? That the E. coli is not able to produce the enzyme citrase, whereas the Enterobacter aerogenes is having the capability of the formation of the citrase. Okay, like here we have discussed, like the E. coli is having the capability of producing the tryptophanase, whereas Enterobacter aerogenes is not having the capability of the production of the tryptophanase. So what is happening here? The citrase enzymes can not be produced by E. coli, but it can be produced by Enterobacter aerogenes. Okay, so means this type of reaction, the conversion of the citrate into oxaloacetate and acetate, and further the conversion of oxaloacetate to pyruvate and carbon dioxide can only takes place in case of Enterobacter aerogenes. Okay, so whatever the sample that we have brought to us and we want to identify and differentiate the bacteria of Enterobacteriaceae, what we have to do, we have to cultivate that bacteria in a culture media that consists of Simon citrate agar media. Now this is one of the specialized media that consists of citrate as a source of carbon. So if only the citrate is available as a source of carbon, then what will happen? Both the E. coli and Enterobacter aerogenes will try to consume that citrate as a source of carbon. But we have seen that this citrase enzyme, for the utilization of the citrate is necessary. But E. coli is not able to produce the citrase. So who can produce? Enterobacter aerogenes can produce the citrase enzyme. So what will happen in case of the Simon citrate agar media, when we go for the inoculation of the bacteria, only the Enterobacter aerogenes is having the capability of consuming the citrate, and it will undergoes the production of oxaloacetate, and further this oxaloacetate is converted into pyruvate and carbon dioxide. But along with the Simon citrate agar media, in the same media we have also added few quantity of bromothiamol blue as an indicator. Now this bromothiamol blue is having some special property that it is green at the pH of 6.9, whereas it is blue at the pH of 7.6. But what will happen if the citrate will be utilized in the media? Then because of the utilization of the citrate, the alkaline end products are generated. And because of the formation of the alkaline end product, the pH of the media will start shifting towards the alkaline side. Initially, the pH is around 6.8 or 6.9, but because of the utilization of the citrate and because of the formation of the alkaline end product, this green color will be converted into blue color because the pH will be elevated from 6.9 to 7.6. So at the pH of 7.6, the bromothymol blue will have a blue coloration. But why this pH is elevated? This pH is elevated because of the utilization of the citrate. And who can utilize the citrate? It is only the Enterobacter aerogens that can utilize the citrate. So again, whatever the sample that is brought to us, if we want to go for the identification and differentiation of two categories of the bacteria, then what we have to do, we will try to cultivate, we will try to grow that bacteria in a culture media that consists of Simon citrate agar media, and it is also having the presence of a indicator that is bromothymol blue. And after the incubation period, if we can observe that the color of the media is converted into blue, then what is our conclusion? That in our sample, the bacteria that is available is Enterobacter aerogens and it is not the E. coli, because E. coli is not having the capability of utilizing the citrate and converting the citrate into oxaloacetate. So by this way we can easily differentiate the presence of different categories of the bacteria which belongs to the category of Enterobacteriaceae. So these are the 4 tests which are routinely performed in the laboratory for the identification and differentiation of bacteria that belongs to the category of Enterobacteriaceae, especially your Escherichia coli, that is E. coli, and Enterobacter aerogens. When we perform the indole test, if the red ring is observed, then we can say that in our sample E. coli is available. If we perform methyl red test and initially if the red color is observed after the addition of the methyl red dye, then again we can say that in our sample E. coli is available. But if the red color is not observed and we are continuing Vogus promiscuous test and then the red color is observed, then we can conclude that in our sample Enterobacter aerogens is available. And after performing the citrate utilization test, again we can conclude that in our sample E. coli is not available, but it is the Enterobacter aerogens.